Thank you. Okay, so without further ado, I will say that I began, my horseshoe crab exposure when I started working at the U.S. Fish and Wildlife Service office in Falmouth, the Gulf of Maine program. And at the time, we were initiating this mapping study of critical habitat for species in decline, and horseshoe crabs was the least appealing species to my supervisor. And of course, it's the first one he gave me. And for the next 2 months, everything I turned into him, he said, no, keep reading, keep reading. Well, if you want to learn science, Wikipedia is a perfectly respectable place to start for a lot of, life science things. And then I encourage you, if you want to go deeper, or if you really want to get in the nitty-gritty and you're willing to read paper abstracts, go right to Google Scholar. So I started on the Gulf of Maine habitat analysis, and that led me to horseshoe crab spawning surveys that we started in 2001, but they went from Cumberland up to Taunton Bay, and we had all these different sites, like 10 or 12 sites up the coast. Every site had a different team of volunteers, and every site had a different tide table, and everybody had training. It was fabulous, and it's full of challenges, but we learned a lot. And from there, I went on. I also, at the same time, did a very detailed tagging study up at Taunton Bay. Taunton Bay, for those who don't know, is the bay behind Frenchman's Bay, which is behind Bar Harbor, Bar Island. Then I also did horseshoe crab scouting surveys on the coast of Maine, and I got to know a lot of people in the community. I actually had somebody have to pull his book out of publication to rewrite a chapter after I did this, which I was pretty happy about. And we took DNA from different sites in Maine to contribute to population studies for the whole species and compare how isolated for how long. Then I went and did more graduate work in Great Bay, New Hampshire, which is a different habitat, but interesting. Okay, so this is going to be pretty much about where, what, when, why, and how. And do they bite, pinch, or sting? And along the way, of course, everybody wants to know, can we eat them? Next. There are 4 species of horseshoe crabs living worldwide, and they have not changed very much in the last 445 million years. We have 1 species in the Atlantic, and that's Limulus polyphemus. The range is from Maine to the Yucatan Peninsula. And then there's a species in Japan and 2 species in Southeast Asia. And in the course of my work at the Fish and Wildlife Service, I started calling around and doing some just follow-up with marine labs and such associated with the universities, and identified that Taunton Bay is the northern extent of the range. And then in the south, it goes all the way down to the Yucatan Peninsula. And interestingly, when you get to extraliminal populations, these are the ones on the ends of the range, they're smaller. And in the middle, they're larger. Who knows why? So, when is a crab not a crab? When it's a horseshoe crab. They are arthropods, so they have copper-based blood. They grow by molting. They are in the subphylum Chelicerata, so they have all these walking legs. They have 5 pair of walking legs, but then they also have these things that look like legs that are little pincers that help them put food into their mouth, and those are the chelicera. And then horseshoe crabs are merostomata, and they have, I'll show you flattened book gills later on my display table, and they have a tail. So unlike the spider, scorpions, and no, that was good. Unlike the spiders, scorpions, and mites, none of them have antennae. And spiders actually have book lungs, horseshoe crabs, book gills, but I digress. Okay, next. So how do we have any kind of, how do we count on horseshoe crabs? And the answer is that horseshoe crabs are an important research species, and they're also important to the biomedical industry. You know those dissolving sutures when you have surgery and they do the inside stitches that dissolve? Made from horseshoe crab shells, which are chitin. If you've had injectable drugs, if you've had surgical implants, all of those are checked for purity with a product made from horseshoe crab blood, and that's called limulus amoebocyte lysate. It was discovered at Woods Hole in the mid-1950s by a gentleman named Bangs. Next. They're also important to the eel and conch fisheries, a little bit of live aquarium pet trade. But ecologically, they're really critical too. Ecologically, they're really critical to spring shorebird migration. And over on the right side, we have red knots. And red knots start in South America. They fly days at a time until they have expended their bodies. They land. They eat for 2 or 3 or 4, well, not even 2, 3, 4, maybe 5 days until they get enough protein and fat to rebuild their muscles and their body fat and undertake another leg. So for us, that would be like go out to the potluck supper, eat, and walk to Chicago before you get your next meal. So they're remarkable birds, and they really count on getting to eat the horseshoe crab eggs that are dislodged by large mating congregations, aggregations. This is mating in the mid-Atlantic at peak season. It's not like that here. We have fewer horseshoe crabs, but we do have more than they thought. And there's sort of this tug of war that goes on between shorebirds and horseshoe crabs, right? So horseshoe crabs bury their eggs a little deeper, shorebirds evolve slightly longer bills, horseshoe crabs need to get their eggs a little deeper, and this just continues to evolve. And so successful nesting is just about the depth of the adult bills of these shorebirds. Otherwise, shorebirds have to rely on eggs that are dislodged by more horseshoe crabs coming in. Next. Historically, they were used for fertilizer. This is also from the Chesapeake Bay area. This is a UNH professor's wife in the 50s on summer vacation. This is Mrs. Milne. And all those horseshoe crabs stacked behind her are drying for fertilizer. And then over to the right, you can see they load them into these cars, slide it down the rail, put it over a freight car and dump them into these freight loads, and they would haul them off to be ground up for fertilizer. So the pressure on horseshoe crabs for fertilizer decreased in the 50s when petrochemical fertilizers became more prevalent. But in the meantime, biomedical kind of stepped in. Next. Okay, they are safe to handle. They don't bite, pinch, or sting. When you pick up a horseshoe crab, please pick it up by the rounded part. This one's, please don't fall apart on me. Not by the tail. And I'll have a little bit more to show you at the end here. They're remarkable for their vision. They have on the tail, they have this array of 24 or more light sensing receptors. They have a compound eye here and on the other side. They don't see very well with their compound eye. If you're out there in hip boots and you're walking around, they'll see the toe of your boot. It might be another horseshoe crab. I might just go over there and see if it's a horseshoe crab, and maybe I want to mate with it. So they'll come by and check your boot out, stroll on, keep going. Then they have these simple eyes, here and here. They even have eyes on their underside, a pair. And so it's, the compound eye doesn't have great vision, but they obviously, light sensing is really important to them. And one of the reasons is that at night, they have the ability to amplify ambient light 10,000 times. So if this room were completely dark and you light one match, a horseshoe crab would see that as if we not one birthday candle, but 10,000 of them. So they can see as well at night as we do in the daylight. Next. So in New England, our population north of Cape Cod is in what's known as the marine boreal zone. Our continental shelf is very narrow, and it drops off. Reminds me of that cartoon, I've fallen and I can't get up. And so they stay in our estuaries and they move to lower areas in the estuaries for the winter. South of Cape Cod, they use the estuaries in the summer and low energy shorelines, and then they go offshore to depths of pretty much up to 100 feet for the winter. And they're picked up in trawl surveys. But, you know, the Chesapeake Bay, the mid-Atlantic, has a really wide, flat continental shelf. Can you think of other differences? I mean, our tide at Taunton Bay, our tide some days is 13 feet. In the mid-Atlantic, their tide is more like 5 feet. When you get to Florida, high tide is 1 foot. So this affects all of your mating strategies if you're a horseshoe crab. Next. Okay. I'll come back and talk more about egg development and why the tides matter, because everything is different in Maine. And I was at a scientific conference, and at the opening social hour, somebody came to me and they said, look, the only environmental variable that matters is temperature, period. And I was so glad that person did that, because I was able to really prepare my talk and show that there's a difference between the mid-Atlantic, south of Cape Cod, and north of Cape Cod. So they did me a favor because I was well prepared and I explained my situation and my data. So what do they eat? Well, horseshoe crabs, they sort of troll around at high tide. You'll see them patrolling and looking for anything they can find: invertebrates, juvenile shellfish, worms, vegetation, detritus. And one of the things that they perform as an ecological service is that they burrow through the sediments. So they keep those sediments turning over. So it's another contribution that they make. This is a bloodworm we found. It was the largest one. It was the only one we saw, and it was also the largest one I've ever seen. So I thought it merited a picture of its own. Okay, next. How many of you have seen children's books about horseshoe crabs? What have you learned about horseshoe crabs from these books? When they spawn, right? Tell me what you've heard. That they spawn at the high tide, right? And that they spawn at night, right? Well, this is Maine. We dare to be different. And the conditions are different here, so that's why. This is an older children's book, The Crab from Yesterday, and you see this boy pulling his wagon down to the beach, and he's going to catch horseshoe crabs and take them and sell them to the fishermen for bait. And he makes 4 cents, and at the end he feels badly and throws his 4 cents in the ocean. Crab Moon is about how you go out at night during the full moon to see horseshoe crabs. And then the last two are more recent additions. I love this high tide for horseshoe crabs because you can see that white disc, and that's a U.S. Fish and Wildlife provided research tag. So that one has a number. And then another great contribution is Horseshoe Crabs and Shorebirds, the story of a food web. So I'm hearing that some people are already seeing horseshoe crabs in Maine. The best time to see them varies a little bit by our winter. So as I've heard tonight, they're already over in Freeport at Winslow State Park, right? And this is very early because we're way ahead of Memorial Day weekend. The full moon was a week ago. So they're really early. So and as I have modeled horseshoe crabs, I had to come up with a surrogate measure because we have boom and bust years, right? And the fact that horseshoe crab spawning has already started now in southern Maine means that this is a boom year. And a boom year is when it starts before the end of May. So spawning is this will be the first two. So it'll be this lunar phase this week, and then it's really a 12-day cycle. There'll be 10 or 11 days, a couple days lag, and then we start building towards another one. It's not a perfect 14-day cycle. Lunar phase matters with the onset. In Maine, it doesn't matter after the initial spawning starts. And remember what I said about all those light sensing spots? If you're a horseshoe crab, you need to know the first lunar phase. So they're cold-blooded. There's a combination of water temperature has to be 69 degrees Fahrenheit or better to get a lot of spawning. And then also what I said about the tides mattering. If you're a horseshoe crab and you lay eggs in Maine and they put them at the high tide line, that sediment, so the way they spawn is the female burrows in, she deposits eggs, a male clamps onto her kelson or tail, and then, and then he sheds milt, and she fans that milt across the eggs. When you see a satellite group of males like this, in fact, the paternity is better for the third and fourth ones than it is for the first male. I'm sorry, it's better for the second and third males than it is for the first male, because the female has this inhalant current, and then the milt is fanned across the eggs. So there's greater paternity by the first two satellite males to arrive.
So can I ask a question?
Yeah.
So Sue, the 69 or warmer, we don't get that this early, do we?
We get it in the shallows.
In the shallows, okay.
I mean, in the shallows, because remember, these are estuaries. There's mud. It's black. It's organic. There's a lot of oxidation going on, and things are burning up or changing, and we get a little bit of activity showing up at 14 degrees, but you don't get big counts until you hit 15 or 16. And then the other thing is that horseshoe crab spawning is much better on a beautiful, bright, sunny day than on a windy, cold, rainy day. Not just because if you want to meet somebody special at the beach, you go on a nice day, but also because a bad day corresponds to wind and waves knock them over and tumble them. And that's just not very conducive to good spawning results. Okay, so eggs need moisture development and warmth in order to move forward with development. And horseshoe crabs in Maine, so the eggs are buried in the sediment 2, 3, maybe 6 inches down, depends on where you are and how much sand there is. If there's too much silt in the sediment, too much clay, it covers the eggs and it interferes with oxygen getting through to the eggs. So what you want is sand, you want warmth to move development along, and you want moisture so the eggs don't dry out. In Maine, if we have a 13-foot tide, then those eggs that are laid at the high tide line, they won't get wet again until the next full moon. And they stand the possibility of not developing because they dry out. So eggs that are laid a couple days after the full moon, when you have maybe 11-foot tides at that end of our horseshoe crab range, those eggs will get moistened more frequently and they have a better success rate. Okay. Eggs hatch in 2 weeks or more. They grow by molting. There's some lovely pictures here of horseshoe crabs molting out of their shell. And so what happens is that molting is controlled by a hormone ecdysone. and they split here at the back of the shell, and then they come out. This is actually the head. So this is the head, the prosoma, the midsection is the opisthothoma, and the tail is called the telson in horseshoe crabs. So they come out of that shell, they swell up, and the shell hardens in a matter of hours. Males don't reach sexual maturity until 9 years and about 15 molts. It's more physiologically costly to be a female. Eggs are more expensive than milk. They have more protein and fat in them. And so females take longer to mature. So females need 10 years and about 16 molts. After that, one of the things we learned was that on the tagging study, we gave everybody individually numbered tags. And after about 7 years, we were only getting 1 or 2 females from the first year class. And then they were gone. By the 8th year, we didn't see any more of those year 1 females. They might have died, not likely. What's likely is that they molted and they got larger. Males started dropping off a year later. So that's something we didn't know. But if you have an animal that requires 10 years to reach sexual maturity, that's really important to know. Because if you overfish, then they can't replace the population quickly as if, say, they reach sexual maturity at 5 years. And of course, wouldn't you know, the females on which the whole population is more dependent take longer. So that's an important thing to know. I will say that adult females are a little larger than males. So here is a picture of the underside of a mature male. That first set of walking legs in males become claspers. And what they do is the males, so the first one there is the female, she's a little larger. The second one is a male. And he holds on to the female at the base of her tail. If I pick these up, they're not very intact. So he will actually clasp on here, and just to be close at hand to cast blessings upon the eggs when the time comes. And he'll actually, it's kind of, I liken it to docking clamps on the Starship Enterprise. The relaxed position is clamped, and they have to consciously unclamp to release. Okay. Next. They're remarkably tough animals. So the shell on top, you can see that big hole. It looks like somebody accidentally dropped a pipe right through it. And you can also see, I mean, we found it as a shell, so it had died. But you can see that it had healed around that hole and survived for some remarkable amount of time. They also, one of the things that we learned on this study is that I think I'm not sure what, it might be seals will come by and take a bite. So on this horseshoe crab on the bottom, it's missing most of its walking legs. And what we saw was, if you looked, you could see where the yellow arrow is. There are little tiny leg tips that are regenerating. So those terminal joints can regenerate. I am misquoted on Wikipedia. They don't regenerate their whole legs. I'm only claiming that they regenerate the terminal knuckle forward, the terminal knuckle outward. And initially, there's a picture up there. It just looks like a little dot. So that was an exciting thing to discover. I've talked about their light sensing capability. And I think I've talked about their habitat use. So I also studied horseshoe crabs in Great Bay, New Hampshire, year-round. We put sonar tags on them. And in the winter in Great Bay and also in Maine, what they do is they go down to the lower estuary to sort of a deeper pocket, and they just stay there. Then they don't really move all winter. But as soon as the water temperature hits 10 degrees C, I was watching them, and I was out there with I had sonar tags on the horseshoe crabs with different pinging sounds and pinging patterns. And I had 29 horseshoe crabs marked, actually 29 that we kept track of, maybe 39 overall. And so we knew when they moved because we had sonar buoys that tracked them whenever they moved. And all of a sudden, when the water temperature hit 10 degrees, it was as if they yawned and stretched and they all started moving upstream. The only ones that didn't were ones that were in a deep channel, and they got a little confused and they moved to a shallower plateau downstream. But they all moved to shallower waters. So they move up into the intertidal flats and they wait, and they get tidally rhythmic. Horseshoe crabs alternate between being diurnal or being tidally rhythmic. So why would you need to know about the tides? Well, if you're going to spawn at high tide, the value is that you have to be there and be ready. And you also need to get out of there and get back to deeper water when the tide starts to drop. So they have these biological clocks, as every living animal that has been studied has. And they go from being circadian to being tidal. And then once they spawn, when they're spawning, they get right up into the upper flats and the shallows. And they just retreat to a little below the low tide mark during spawning season. And then once spawning season is over, instead of being all the way up here in the upper parts of the bay, they kind of drop down a little bit. And then they're in sort of foraging flats. And as fall starts to happen, they start to shrink their range, get a little deeper, a little further away from the shallows, because the water temperature is more stable in those quiet, deep holes in the winter. All right. What did we learn from tagging? Well, the top frame are the counts, how many animals we saw each day, and this is in 2001. And that compares to the second frame are the lunar phases. The dark one is the new moon, and the one that's yellow is the full moon. So you can see that spawning doesn't occur in Maine on the moon. The peak activity occurs a little after the moon. The third frame down is water temperatures, and then the fourth one is the maximum high tides, or the height of the high tides. And so tidal amplitude matters. The lunar phase matters for the onset of spawning, and it's also not a straight 2-week, 14-day cycle. They have a plan, and we're still working on it. This one looked at weather. You can see that the dots all the way over to the right are days that we had beautiful, bright, sunny days, and all the way to the left here is what happens when it rains, which is very merciful, because if you're fielding a field team and you have to be out there in the driving wind and rain, at least you're only out there for a half hour. Next. And these are examples of the tags that we used. I have a few examples here. These are what I call spaghetti tags. They might look familiar. They're double T-bar tags, and they're made from clothing tags by a company in Washington State. These have a number and an email on them.
Do you get returns?
Not after 7 or 8 years.
Yeah, but you do get.
I did get some returns.
Yes.
And the interesting thing what's interesting about horseshoe crabs in Maine is that whatever we learned about horseshoe crabs here, yes, the environmental cues for spawning are different, but we don't have horseshoe crabs harvested in Maine for biomedical use, and we don't have horseshoe crabs harvested for bait. So we have the only natural population on the East Coast that's a large functional population. We have normal ratios of males to females. Whereas in the mid-Atlantic, it's very difficult to do population dynamics when you have 4 times as many males as females, because the fishermen like the females as bait. They cut them up, and the mucus on the eggs has pheromones in it, and the fish like them because there's a horseshoe crab at the end. And so their population ratios, their sex ratios are highly skewed because of hundreds of years of harvest. These tags I was very careful to pick tags that would break off. So you can see the middle one on the right, that's a horseshoe crab with a tag that broke off. And this one on the lower left is a horseshoe crab that molted out of a tag. This piece of the shell is called the genal angle. And it's basically when we have to drill a hole with a drill, and then we put the little tagging gun through. It doesn't strike any living tissue, it just strikes open shell. And so we almost never get any blood. And if we did get blood, it's a couple of drops, and we put the horseshoe crab back in the water, and that coagulates instantly. So the goal of the tag was to not affect survival of the individuals, and we were successful at that. Next slide. This is a little bit about the spawning surveys that were done. We collected data. This is sort of the standard data plan. People like to tell you, and I like to know about every horseshoe crab you found, right? But the statisticians, they want something they can measure. So when we did this tagging study, we had sections that were 10 meters long with a flag at each end, and we kept 2 columns of data. And one column of data was how many horseshoe crabs were within 1 meter of the shore, and the other one was everybody else you saw. If you've done things like this, so if you go out in the woods and you're a great birder, right, and I go with you and I'm a terrible birder, and we're looking on opposite sides of the path, the great birder is going to see a lot more than the novice. And so this system of having 1 meter and everything else allows you to reduce the bias that's in your data and make it more robust for statistical analysis. Enough of that. I think I've covered everything here. And that's all I have to present tonight.
There we go.
So.
I have questions.
Thank you. Questions?
So I have. I'm sorry, everybody, but I'm really interested in this. So, does the state of Maine have any management? It's prohibition for commercial harvest.
Okay, so the state of Maine has what's called de minimis. Okay, so the state of Maine doesn't have any commercial harvest, and there are no blood facilities. By the way, it is incorrect, as stated in different websites, that you can drain all the blood out of a horseshoe crab and throw them back in and they'll magically survive. They won't, any more than we would if you drained out all of our blood. So, that's a mistake. But they take about a third of the blood, and it depends a little bit on when the flow rate slows down, and also on the health of the individual animal that before they collect blood as to what the survival rate is. Some labs say they lose 5 to 7%, and other labs have been credited with losing up to 25% of the animals that were bled. There's a lot of management questions. One of them is, if you're going to bleed animals, then make the bait fishermen take their quota from the animals that have been bled, and you'll reduce the total mortality on the population. They didn't ask me, but...
Sue, what about like bycatch? Bycatch is the stuff that's caught that you weren't fishing for. So if you're fishing for mussels, dragging for mussels,
Well,
Are they, what happens if they catch me with a bunch of horseshoe crabs?
well, truthfully, you know, there's first of all, enforcement would have to be happening.
Right.
De minimis means you can take up to 25 a day. However, Department of Marine, State of Maine Department of Marine Fisheries says that you have to have a permit to pick horseshoe crabs up even, okay?
Right.
So if I wanted to bring a live horseshoe crab to this talk, which I considered, but I learned my lesson last week that if you hand it out, people don't pay any attention to what you're saying after that. But I would have to get it from somebody who had a permit to have that. That said, you know, people are like, I have all kinds of volunteers on my crew, on my tagging crew. I had somebody who is a dedicated environmentalist without any science background. And she would go out there with a horseshoe crab and she would want to do a salmon release on it. And I would be like, we need to keep moving within the time frame to get through the transect before the tide drops. And it's an exoskeleton. And so I would be perfectly happy to gently frisbee them 4 or 5 feet out into the water. She did not take this well. But, and then, I mean, I had this wonderful woman who was she's a faculty member and she worked in the music department I think at Oberlin in Ohio. Our best data recorder ever. She never made a mistake. And then I have somebody who, whenever somebody saw something odd that was unusual or say, oh, he's missing a leg, there was always a mistake on the next record because she was distracted. So, you know, we got actually something like 15,000 observations, about 85 or 9,500 individuals. And the folks in U.S. Geological Survey who do the statistics and the population management and the genetic studies, when I mentioned that we had that many, he said, oh, I never thought you would have that many. So there's a very robust population in Taunton Bay, which is great. And also when you consider that that was just our 350-meter piece of shoreline, never mind the rest of the 6 coves of the bay. So that was pretty exciting. Handling, so technically you have to have a permit to handle. Yeah.
Great.
Any other questions?
Can
Yeah.
you take home a shell?
You can take home a shell.
Okay.
Yeah. But, and you know what? I'll just tell you, these shells, for instance, now this one died. And we know this one died, well, if it weren't 10 years sitting on my book, as opposed to molting. We know it died because there's no crack here around the seam. This one molted. Oh, well, this one molted and it completely cracked and separated. Okay. And you see how it just forms there's a little dip there. Okay. This is the underside. So here is this is the head, the prosoma. These are the little chelicera, the tiny little feet that help them feed themselves. This is the mouth. These are the walking legs. First walking leg in a male, first pair of walking legs in males become claspers. One side, they actually form these little boxing gloves like itty-bitty lobster claws. And then the last pair are these swimmerettes. That's how they get along. And these are their book gills. So it's true they're not going to win any beauty contests. It's also true that I was quoted, I don't think I said this, but by the Bangor Daily News saying, I love them, but there you go. They're fascinating, and there's a lot to know about them. And, you know, how do you predict a boom or a bust year? And I happened to talk to somebody at a program, and he said, you know, this is Dave Smith. He's trying to do population estimates and how quickly populations replace themselves. And, you know, he's in charge of that for the whole U.S. Atlantic States marine fisheries. That's part of his wedge of the pie. And I said, I'm working on boom and bust years. And he said, yeah, I can't figure that out, but I have figured it out. And the answer is, you know how your oil gets delivered? By heating degree days. Heating degree days is a measure of how the weather warms. So you contact NOAA in Gray, and they will send you heating degree days. If you're trying to do light studies, I had this wonderful several years of light studies, and the data weren't very conclusive. And after collecting data for several years, I discovered that the tanks were located outside the greenhouse at the marine lab. And when the janitor comes in at night, he turns on the greenhouse lights and they stay on all night. And so that data was worth trash. It was worth nothing because, you know, and you never know that, right? You have no way of knowing that janitor's doing his job. I'm a research student, I'm doing my job. And until I happened to come to do overnight counts in the spring at the end of 3 years, and I was in there at 3 in the morning, and the place is lit up like a mall. And I was like, I don't think the tank data is any good. So, you know, as John said, horseshoe crabs are not as exciting. Horseshoe crabs don't even get as much funding as dinosaurs. Dinosaurs are a little better funded, and there's still a lot to learn. So.
And I'd just like to add that, you know, the life of a field biologist is pretty grueling. I mean, you have to put everything on the back burner and dedicate yourself to that calendar and the temperature that Sue was talking about. And, you know, and she's done this for what, 20-something years?
Well, really, the hero of this story is the family that hosted me in Taunton Bay. Yeah. I was hosted by 1 year by somebody, and the next year by somebody else, and the next year by kind of a patchwork. And then after that, the Harrington family hosted me for the next 7 or 8 years. I mean, I watched their daughter grow up. And I will tell you, it was terrible on all of us when I was doing edits for a paper, and I would send them out. I mean, 47 edits to my advisor, and it was grueling. But yeah, they were wonderful. They hosted me for a very long time, and after a while, I graduated to. Well, sometimes I had a house, and other times for a number of years, I had Shea Shasta. So they had this wonderful, out of the 50s, Shasta camper with birch paneling, and it sat over by the garage and had Christmas lights inside, and it was delightful. So yeah, I applaud the Harringtons, and the Friends of Taunton Bay paid expenses. Conversely, when you're working with volunteers, almost all the volunteers are great. There was one day when I said, I'm really tired, I think I'm ready to go home overnight. And somebody said, oh, I'll do the count tomorrow. Well, tomorrow came, and she was downtown and decided, there won't be many today. And I was like, I would have stayed and done that, you know.
It doesn't work. It takes real dedication.
I mean, you know, but that's year 10. So my sister shows up to visit. We have to go for a hike. She gets lost, and I called her on the phone and said, I am leaving. I have to get to a count. And she was like, I can't believe you would leave me. I'm like, you just don't understand. You know, this is 10 years. I've been out here every month of June for all these counts, and I've never missed one. And guess what? You're second fiddle. Get back to the house on your own. You have your own car. I have my car. And, you know, the volunteer surveys up and down the coast went great. There was just one day when somebody had guests who showed up and they went to lunch, and it happened to be one of the highest count sites in the state. And it was right the middle day of the peak week of the whole season. So, you know, I'm demanding, and we get results. And so I just think that if I were to do that again, I'd have to have double teams to make sure that somebody always had like a person they could call as their backup. So But anyway, and statistically, you just take the average of the days on the other side of that. But I really wanted that high count that I was convinced was there that day. So.
And Sandy.
The fishery down on the Chesapeake area, is that limited to what they can catch? And, you said there was a difference between male and female, but is there any limits that they put on harvesting?
There are landing limits, and I'm not sure what they are for males and females. I have a funny feeling that they don't. I think it's just the number, or it's either number or pounds. But it's not male or female because the only way you'd verify is by looking at every one. So that's why I think that's not the case. My models here. This is a horseshoe crab this is a horseshoe crab from Delaware. And you can see this is a big horseshoe crab, okay? And, I spent a few years waitressing, and I can stack 6 Maine horseshoe crabs up my arm like they're plates. But I can only get one of these because they're just too, they're just bigger. You can't quite balance them and they don't stack well. This is probably this is a female, and even though we don't have the underside of this horseshoe crab, we know this is a female. Well, we think it's a female because there's a little wear on the shell here where she was clasped by a male during spawning. And there are some juvies here. So you'll see horseshoe crabs in August. You'll see lots of these shells. And if you turn them over and look, you'll see a little split on the underside basically along the forehead. And that's a molt. So you are welcome to come handle these. Here's one. You can just sort of press it a little bit.
And they come out like jelly.
Yeah.
Pretty much.
They're kind of, I haven't seen very many. I love the question that when people say, what do horseshoe crabs eat? Well, when I was at UNH, we had these tanks in the marine lab, and one of the lobster guys was there, and I said, hey, Chris, I think one of your lobsters died. And he goes over and he looks, he says, oh no, that's a molt. The lobster split down the back. You know how you see little lobster shells on the beach and they're split down the back? Well, that's because they molt and they split down the center of the dorsal surface. And he said, oh, you saved that one. So he puts this lobster into the tank with the horseshoe crab, takes it out from the lobsters, puts it in with the horseshoe crab. Well, what do you think horseshoe crabs eat when they can get it? Lobster. So that lobster was in there, and those horseshoe crabs just went up to that lobster and just folded on it and just chewed a little. And when they'd eaten their fill, they let go, and the next one came along, and they ate until they were done. And so the answer is they eat lobster when they can get it. But, they don't get it very often.
So I know in the Delaware Bay area, the red knots come in and feed on the
Yep.
eggs. That I don't see that happening in Maine. So what preys on the eggs? And
Well, shorebirds do, but it's kind of a skip. We don't have the densities that they need. So if you're out during spawning, you'll see these little shorebird holes. If you get out there a couple hours before the tide comes up and obscures them, you'll see the holes. And they're also, you can see nests because the female will burrow in, and then when she's done, she goes out, but it leaves a little depression. But it's pretty obscure within a couple of tides. Probably around 2003, I was going to a meeting on the Cape, and I went to the beach for a day with my nephews and niece, and they're down there digging in sand, and they're digging like 6 or 7 or 8 inches down. And Alex comes up and says, Auntie, what's this? And he had opened up a nest of horseshoe crabs. And this is just an experiment I would love to do. So horseshoe crabs have all these larval stages that look like trilobites. They're called instars. So they make, in the first couple weeks after the eggs hatch, they go through instars before they look like itty-bitty horseshoe crabs. That nest had all these itty-bitty horseshoe crabs that were not as large as my pinky nail. That includes with the tail sitting on my pinky nail. They look just like horseshoe crabs, but they're this tiny. And I think that what happens is they're in that nest. They eat whatever's in the sand. They probably eat eggs that didn't hatch. They probably ate eggs that hatched after they did. And then you have this nest that's probably about like a 5 or 6 inch ball. And it's all these little greenish, semi-translucent horseshoe crabs. And how do they get out from under 8 inches of sand? Well, you wait till the tide comes in, right? And when the tide hits that level again, and it's high tide, the water comes through, and then they can swim up through the sand. And the only way to document that would be to have a tank with a pump on a tide cycle and a camera that just sat there and got the day that they all decided they were big enough to swim their way out of the sand. Patty Crooker over at Thomas Point Beach, when she was alive, told me that her father hated horseshoe crabs. And they had made that beach. They couldn't make it now, but they had, because they owned gravel pits, sand pits. And they made that beautiful beach at Thomas Point Beach, which is a great place to go see horseshoe crabs. And he would go out at night and dig a big hole with a piece of equipment, put all those horseshoe crabs in it.
Oh, no.
Well, they make a living by burrowing through sediment. I'm sure they were back in a couple of days. And, you know, we've done some weird things with horseshoe crabs. Like, I was working on horseshoe crab activity levels, so I had accelerometers on them. And I was setting them up to put them in these cages that we put out in the estuary. And I was, you know, the lab manager said he would take me out to set my gear, but we had to hurry because he had to pick up his kids at daycare by 5. And so I'm quickly getting all my little motion detectors glued to the backs of my horseshoe crabs and getting pingers on them so I know who's who and where they are. And I get all set to run out to the boat, and I'm missing one. So I make up another. Well, we're in a lab, and when you leave horseshoe crabs on a flat surface, they start going around in circles because they're looking for the slope to get back to the water. So I pull out a drawer and I park them, right? I guess somebody else was pretty surprised on Monday when they opened the drawer. And there was a horseshoe crab. They were like, hello. They do pretty well. They can survive, you know, they can survive being out of the water, not indefinitely. Their survival is better if they're right side up. But, you know, they can be kind of fry-brained, and you can just take them out of, you know, you might see them on the edge of the shoreline. Just take them and put them in the water and sluice them around a little bit to bring their temperature down, and they perk right up and leave. So, so they don't bite, pinch, or sting. But I will tell you, if you pick one up and you pick them up here, right, they will close their shell like this, and they have movable spines here. So if you pick them up and you're afraid of the head of the animal, you can get pinched. But the walking legs don't pinch, they don't sting, the tail doesn't sting. So the way to pick up a horseshoe crab is you grab it by the rounded head part. And that is after you get your permit from DMR to handle that. So, and if you're at Thomas Point Beach and you see people going, ooh, and grabbing them by the tail and throwing them, just pick one up and explain to them. And you can really be an ambassador for horseshoe crabs now that you've been through the training. So, Lynn.
So, one more question. So I've noticed down at Cove Reserve, it's a lovely sandy, beachy area, and I've seen the shells there. So I think that might be a good place to go look for them. So I'm just, taking into consideration all of your conditions, would you want to hazard a guess at a peak day?
Sure. I would say now you can go anytime during the daylight high tide and look at the tide chart. They won't be there on the lowest tides, and they won't be there on the highest tides. Okay, you'll get them at high tide, but they need to be mid-range. And you'll see them this is an early season, so you'll see them for the first, no, not many after the first 2 weeks of June. I kind of think that those are the juveniles that are coming in to the population that are coming late. You know, they're kind of just getting the concept. So, thank you all for coming.
Thank you.